Hello students, welcome to Distinction Learning Platform, and on this platform you'll find revisions, expansion to difficult topics, a lot of stuff. So before we begin our lessons, make sure that you subscribe so that you can be updated with all upcoming videos, and there is a link in the description that will take you to the website where you find now other subjects like English, biology. Maybe if you are taking university courses, they are also there, so don't miss out on that. Make sure that you click on that link. Okay, now to make the story short, in this video we're talking about enzymes under biology. Okay, so now let's explore more about enzymes. So we talk about enzymes, enzyme classification, characteristics. We talk about the factors that affect the enzymes, the rate of enzyme activity. There are a lot of things that we need to cover under this subject, okay, or under this topic. So now let us begin talking about this. So what are enzymes? From biology, we know that enzymes, these are what we call biological catalysts. Okay? Very, very simple definitions. So an enzyme is a biological catalyst. Now, what does that mean? We need to understand what that means. Why? Because if you look at the human body works because there are some chemical reactions that are happening in our body. And those chemical reactions, for them to happen efficiently, there has to be some certain substances or molecules that helps those chemical reactions to happen, and we call them biological enzymes or enzymes. Okay, so a good example of that chemical reaction that I'm talking about is what we call respiration. For respiration to happen, there has to be what? Certain enzymes that need to characterize that needs to speed up that rate of chemical reaction. Okay, so now enzymes, we are just saying these are biological catalysts. Now another important term that you need to understand is what? A catalyst. What is a catalyst? Because we are saying enzymes are biological catalysts. Now what is a catalyst? A catalyst, they say, it is a substance that speeds up the rate of chemical reaction without itself being changed. No wonder we are saying an enzyme is a catalyst. Okay, so now we are saying, what is a catalyst? A catalyst, we are saying it is a substance, okay, a substance that speeds up the rate of chemical reaction. Speeds up the rate of chemical reaction without itself being changed. Itself being changed. Being changed. Very, very simple definition. Okay, that's what we call a catalyst. Now, so basically, if we put these two definitions together, then that should be the definition of an enzyme. So in short, what is an enzyme? An enzyme is a biological catalyst that speeds up the rate of chemical reaction without itself being changed. Okay, that's the full definition of an enzyme. Okay, now what else are we supposed to talk about these enzymes? So let's talk about the types of enzymes, because we need to understand the types of enzymes so that we can have a clear picture of whatever we're going to be talking about. Okay, now what are these types of enzymes that we're talking about? Okay, so let's talk about these types of enzymes. So types of enzymes or classification of enzymes. Okay, so enzymes, so we're going to classify enzymes into what? Two categories. Okay, two categories. The first category, okay, is according to what? The location. It's according to the location where you can find them in the human body. Okay, that's the first classification that we're going to talk about. So, and the other classification that I'll talk about here on the second point is according to the type of food that they are going to break down. Okay, so now let's start with the location. I'm going to give you a good example so that you can follow on this one. So if you look at the human body, remember that the human body it's going to have the blood vessel. So let's try to imagine to say that's a blood vessel. Okay, so that is a blood vessel. And remember that inside the blood vessel there are some certain cells like red blood cells, white blood cells, and other cells. Okay, and outside these blood vessels, there are what we call cells. Okay, there are cells here. Okay, or the body cells. Okay, so let's try to imagine to say these are what body cells, and this is a what a blood vessel. Okay, so we need to understand something because minus this diagram, you can't understand the types of enzymes according to the location. Okay, so now if you look at this, there are some enzymes that can be found inside the body cells. Okay, inside the body cells, what we call those enzymes? We call them intracellular enzymes. Why? Because they can be found inside the body cells. Okay, so let's try to imagine to say this is an enzyme inside this body cell. So and this enzyme is what we're going to call it what an intracellular enzyme. So we're saying the first enzyme is what we call the first classification according to location, and these enzymes are called what intracellular enzymes. So intracellular enzymes, they can be found inside the body cells. So we're saying can be found in body cells. Very, very simple to remember. They can be found inside what? Body cells. They can be found, inside body cells. Now, remember, if there are what we call intracellular enzymes, basically there are some also enzymes that can be found outside the body cells. Okay, like this one. If this is these are body cells, but the enzymes that can be found outside the body cells, or they can be found within the blood vessels, as long as they are not inside the body cells, we call them, the opposite of intra is extra. So I'm going to say these are what we call extracellular enzymes. Okay, and we are saying just the opposite. We are saying, and these can be found outside the body cells. So we say can be found now outside the body cells. Very, very simple to understand. Now, up to now, let us recap whatever we've talked about so far. So what we are from talking about is the definition of an enzyme. An enzyme is what is a biological catalyst. Okay, and what is a catalyst? A catalyst is a substance that speeds up the rate of chemical reaction without itself being changed. Okay, a good example that I can give on catalyst, we are saying something that's going to speed up the rate of chemical reaction. So I'm going to give you an example of that. So an example is this one. If we have this chemical reaction A, let's just say reaction A. Okay, so reaction A, normally if you put it and you have a stopwatch there, you measure the time taken for reaction A to stop, then you find out to say this reaction A is finishing at maybe after 5 minutes. Okay, then if you add now an enzyme to reaction A, you expect that reaction to finish faster, meaning that that substance that we've added to reaction A, it is like making sure that reaction A is what finishing faster, speeding up the rate of chemical reaction of that reaction A. So meaning that we expect, if you put now an enzyme to this reaction A, then we expect maybe to finish less than 2 minutes. Then that's what we mean if we say an enzyme is a biological catalyst which speeds up the rate of chemical reaction but without itself being changed. We'll understand that last definition, last statement, which is without being changed. We'll understand it as well. Okay, so now let us finish the classification. let us finish the classification. Okay, so now this is according to the location or where you can find these enzymes. You can find them within the body cells, which we call intracellular enzymes, and we can find them outside the body cells, and we call them what? Extracellular enzymes. Okay, now we go to another point. we go to another classification. We can classify the enzyme now according to the what? Like the nutrients they digest or they break down. Okay, so so we can classify them according to the nutrient breakdown or where they act upon. So what type of nutrient they act upon. Okay, we can classify enzymes according to that, and this is the famous classification. Even if you forget the first classification according to the location, make sure that you remember the classification according to the nutrient breakdown or the food molecules that they break down or that they like act upon. Okay, what is that? So we've got different types of nutrients in our body. What are those? Let's start with the famous one, which is carbohydrates. Okay, there are some certain enzymes that act on carbohydrates, and there's a name that we call those enzymes according to what I'm going to say here. Okay, so the first nutrient that they break down is what? Carbohydrates. Carbohydrates. Okay, carbohydrates or starch. And what do we call those enzymes that act on carbohydrates? We call them what? Carbohydrates. Carbohydrates. So that's from this word carbohydrates. You are going to say carbohydrate. But the moment you reach E on A, then you just remove TES. You just remove this T and E so that it becomes what? SE, carbohydrates. Okay. So as you can see, these, so let me highlight this. Okay, the ending part here, AS. Okay, carbohydrates. AS mean that it's describing this is an enzyme. Okay, so those are carbohydrates. So carbohydrates, these are the enzymes that act on what? Carbohydrates. Okay. And a good example of these enzymes that act on carbohydrates, we call them carbohydrates. But examples of these carbohydrates, that's what we're going to talk about under digestion. There is salivary amylase, there is pancreatic amylase. So those enzymes, they act on what? They act on carbohydrates. Okay. So a good example of a carbohydrate is what we call what? Amylase. So let's just pick one, amylase. Make sure that you take down on that. Okay, that's a good example. We go to another type of what? Nutrients. What are these enzymes that act upon? Okay, so we've got again proteins. Proteins. These are nutrients. Proteins. Okay. So enzymes, they also break down proteins. Okay. Now, what are enzymes? What are those enzymes that act on these proteins? We call them what? So as you can see, we're going to follow the same rule here, the way I was writing this one. So I'm going to say pro. But the moment you just reach here, then you're going to put AS. Okay? So that as you can see, AS means that this is an enzyme now. We're going to call it what? Proteins. Okay? Proteins, these are enzymes that act on what? Proteins. A good example of a protease or the enzyme that act on proteins, we call them what? We're going to say maybe trypsin. So this is a good example of an enzyme that will act on what? Proteins. That is what? Trypsin. And don't worry, we are going to understand on these specific enzymes when you reach what? Digestion, or the topic of what? Digestion. Okay. Another important nutrient that we need to be familiar with is what? Lipids or fats. Okay, lipids or fats. So this is a very important nutrient that you need to understand. Now, lipids, they are going to have a certain enzyme as well. So we're going to say lipase. Okay, lipase. Lipase is a good example of what? An enzyme that will act on what? Lipids. Okay, and a good example of these lipases is as lipase. So this one doesn't change. Okay, a good example of lipase is as lipase. Okay, and we need to understand to say an enzyme, they always act on different what? Nutrients. So there's an enzyme that will act on carbohydrates. We call them carbohydrates. And what is a good example of a carbohydrates? We call it amylase. Okay, we'll talk about those. And protein, we call them what? Protease. And a good example of a protease is what? Trypsin. But there are also other examples like pepsid, trypsin. Those are good examples of what? Protease. And the last one is what? The enzyme that will act on lipids, and we call it what? Lipase. Yes, yeah. So now we can proceed. We can proceed here on what? Characteristics. Okay? Characteristics of what? Enzymes. So what are the characteristics of enzymes? So before we go to characteristics, we need to understand what it means when you say characteristic. So characteristics, it means this word, it means something that will help us to identify a thing or something. Okay, so some features that help us to identify a certain substance, and those things, these are what we call characteristics. Okay, if I'm going to ask you to say a good example on that statement that I've told you is if I ask you to say, what are my characteristics here? Okay, you can say a lot of features so that someone can identify me. So those things that you tell someone so that someone can identify me, those are what we call characteristics of me. So also here, what are the characteristics of what? Enzymes. So characteristics of enzymes, it means that features or things that will help us to identify that definitely this is an enzyme. Okay, so what are those features? We know the first feature is what? Enzymes are what? Are catalysts. We know. So enzymes are what? Are catalysts. And that is very, very true. Enzymes, all enzymes are catalysts. That's from the definition we say a biological catalyst. And what is a catalyst? Something or a substance that speeds up the rate of chemical reaction without itself being changed. Don't forget about that, okay? Enzymes are catalysts, okay? Because they speed up the rate of chemical reaction without themselves being changed. What else? Enzymes are protein in nature. So this is also another important feature, protein in nature. Another important of what? Characteristics of an enzyme. Okay? So enzymes are protein in nature. What does it mean? It means that these enzymes, if you look at their biological makeup or the way they are made out of proteins, okay? Because there are some certain substances that are made out of lipids, carbohydrates, but enzymes are made out of what? Proteins. No wonder we say enzymes are protein in nature. So that is a very important feature to talk about or to describe an enzyme. What else? Okay, so what else? Enzymes performs both forward and reverse reactions. Okay, so enzymes perform both forward and reverse reactions. So that is a very important characteristic. So if you get an enzyme, so let's say this is a certain substance, okay, in a chemical reaction, and this certain substance has to be converted into D by what? An enzyme. Let's say an enzyme is what? Let us say. So this enzyme letter C is making sure that this substance A is being what, converted into what, B. Okay, now reverse and forward reaction, what does it mean? So enzymes, they have that ability to convert a certain substance into a certain product, and that product to take that product and back to what, a set like the initial state. What do I mean? So these enzymes are able to take this letter A to B, but also they have that ability to get this letter B and back to what, that is what, forward and reverse reaction. But how can we relate to the real thing that we have here? Okay, apart from just talking about the letters, changing letter A to B and B to A. Now, in real life, we need to understand to say enzyme. So I'm going to give you a certain scenario that you need to understand. So if I get carbohydrates, so I'm going to get a certain nutrients, okay, like carbohydrates. So let's just say C for carbohydrates, Ca. Let's just say Ca, carbohydrates. Carbohydrates, you know that good example of carbohydrate is what? Starch. So you can get carbohydrates and convert this carbohydrate into what? Glucose, according to digestion. Okay, so enzyme, there are some enzymes that will help to break down this carbohydrate and convert it into glucose. Again, these glucose molecules, they can be converted back into what, carbohydrate. That is forward and reverse reaction. So that is very, very important what, thing to understand. Okay, so that's what it means. Okay, and let's talk about this one. Enzymes are what? Specific. Are specific. Enzymes are specific. What does it mean? So make sure that you should be following any point that I'm telling you, because they like bringing during what, an exam. Okay, enzymes are specific. So what does it mean? So this, I'm going to write something here. This is going to follow what we call lock and key theory. Lock and key theory or mechanism. So let's just say theory. Okay, so it was this one was being talked about in a like a long time ago. It was used to describe the characteristics of what, enzyme, this theory. So lock and key theory. So what they say is like this. So these enzymes, they are like lock and key. So as you can see, a key, it has its specific lock. Okay, because you can't get a key and try to unlock another lock. Like, what do I mean? So I'm saying, if you have key A, if you have key A and B, and key A is specific to B, you can't get key A and unlock key C. It is very, very impossible. So key A is specific to B, and if you get this key A and try to unlock this lock C, it is impossible. Okay, so that's what that theory talks about. So enzymes, they are like a lock and a key. Okay, so what does it mean? It means like this now. So now let me give you a real time example. So if you have carbohydrates, okay, and proteins, if I get an enzyme, a carbohydrates, like I get amylase. If you remember, amylase is a good example of a carbohydrate, or enzymes that work based on carbohydrates. If I get amylase and take this amylase so that it can start breaking down proteins, it is impossible. Why? Because amylase only works on carbohydrates. Okay, they are specific to carbohydrates. They can't work on what? Proteins. That's what we call being specific. Okay, and you need to remember something here. I'll write a diagram here, because they like bringing this question. You need to remember it, this one. Okay, they like bringing stuffs like this. Okay, 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 so as you can see this diagram, this is the diagram that describes the locality theory and as well as what the specificity, okay, specific of an enzyme. Okay, so this diagram is talking about these they are like the symbols that are on this diagram. Then they are going to ask you to identify this and to identify B, to identify C, and to identify maybe D here. Okay, which is very simple. As you can see, if they ask you to identify letter A, okay, this structure A, you're going to say this is an enzyme. Why? As you can see, this enzyme before the chemical reaction and after the chemical reaction, it is just the same. As long as through the chemical reaction it is not changing, and that structure is qualified to be what? An enzyme. Why? Because we said an enzyme is not what? Changed. Even after or before a chemical reaction, it does not change. Okay, so that's the characteristics of an enzyme. So without itself being changed, so an enzyme is not going to change from A. As you can see, this structure is just the same up to here. So this is an enzyme. So A is an enzyme, and the enzymes, while they act upon, while they are acting upon, these are what we call substrates. Okay, so B is what? A substrate. So because a substrate is what? Just a substance, substrate, okay? Just a substance where enzymes act upon, okay? Now, so if this is a substrate, substrate, so that is a substrate, then this is an enzyme. Now, what about this one? As you can see, this one, it is more like an enzyme and a substrate is what? They are combining together. They are going to form what we call enzyme substrate complex, okay, or substrate enzyme complex, whatever the word that you are going to what to use. Make sure that you just combine enzyme and what substrate. So enzyme substrate complex. Okay, and what about this one, the D? These are now products. Okay, so apart from this is an enzyme and these are what we call products. So these you are going to be examining on that. So make sure that you know this diagram, you know it deep down your heart. Make sure that you try to remember it. Okay, and this diagram is just coming from what this important characteristic. Okay, now the last characteristic is this one. Okay, the last characteristics is enzymes are affected by, okay, pH. They're affected by concentration, temperature, inhibitors, and cofactors. Okay, that is a very, very important part now. Enzymes are always affected by what? pH, concentration, temperature, inhibitors, and what? Cofactors. These are very, very important points to take note. Okay, okay. So these are the characteristics, or these are the things that are what that help us to identify that this is an enzyme. Now this last important point here, okay, we'll talk about in another video here, or the things that affect the rate of enzyme activity. For now, we'll just talk about them briefly, but in the next video we'll talk about the exactly what happens if pH affect enzyme activity, the temperature affect the enzyme activity, concentration, like that, all those things. Okay.